Good evening, everyone. My name is Sandy Gill. I'm a board member at the Calgary Public Library. It's my pleasure to be your MC this evening and to welcome you to this event, a wonderful partnership between the University of Calgary, Alberta Health Services, the Tom Baker Cancer Centre, the Arnie Charbonneau Cancer Institute, and the Calgary Public Library. In the spirit of respect and truth, we honor and acknowledge Mohkinstsis and the traditional Treaty 7 territory of the Blackfoot Confederacy, Siksika, Kainai, Piikani, as well as the Stoney Nakoda and Tsuut'ina Nations. We acknowledge the members of the Métis community, and specifically the Métis Nation Region 3. Finally, in the spirit of reconciliation, and because we are all treaty people, we also acknowledge all Calgarians making our homes in the traditional Treaty 7 territory of Southern Alberta. The Calgary Public Library gives everyone access to stories that entertain and instruct. During our temporary closure to help limit the spread of COVID-19, we are still a community hub where Calgarians can come online to learn and share ideas through digital resources and virtual programming like this. The Arnie Charbonneau Cancer Institute is a community of researchers and educators dedicated to achieving a cancer-free future. The Institute brings together scientists and physicians to integrate research and care across disciplines, from understanding and preventing cancer to transforming its detection and treatment to improving life with and after cancer. Its members include researchers at the Tom Baker Cancer Centre and the University of Calgary's Cummings School of Medicine and Calgary's teaching hospitals. Tonight, Dr. Jennifer Chan will moderate the discussion with Dr. Erin Goodarzi and Dr. Gwyn Bebb, and then there will be time for audience questions at the end. We invite you to ask your questions via the chat function on your screen, which looks like a little speech bubble. In addition to these questions in real time, we have received questions for the panelists beforehand, and we will attempt to answer as many questions as time allows. I'd like to now introduce Dr. Jennifer Chan, the Deputy Director for the Arnie Charbonneau Cancer Institute. She is our moderator for this evening and is a clinician scientist at the University of Calgary. As a neuropathologist with a focus on oncology, her clinical expertise is in the diagnostic evaluation of pediatric and adult brain tumors, and her research applies concepts from developmental neurobiology to further understand mechanisms of disease in brain tumorigenesis. Please welcome Dr. Jennifer Chan.
Sharing my screen. Hopefully you can all see that. Thank you for that kind introduction. It's a pleasure to be here and to be your host for this evening. So this is the third in our public lecture series that we put on, and usually we're called Late Night Lab, and we talk about different aspects of cancer, share some of our research work, and at least in the previous sessions had some hands-on activities for our guests, hence the lab part of Late Night Lab. But during COVID times, we're now late night Zoom, as you can see. But we do hope that in the future we can meet you again in person and kind of resume our previous format. In this particular session called DNA and Cancer, we're going to delve into some of the biological underpinnings of cancer and also some of the treatment implications of specific types of damage that you can get in the DNA. And to get us started, I'm going to give you a little bit of background information before turning it over to our two guest speakers, Dr. Erin Goodarzi and Dr. Gwyn Bebb. And then again, finally, we'll finish with the question and answer session. That's that. So just to review a few basic facts, about 1 in 4 Canadians is expected to die from cancer, and even more actually are expected to be diagnosed with cancer in their lifetimes. So about 1 in 3 Canadians will actually sometime in their lives be diagnosed with cancer. So this is actually quite a common disease. So over 80,000 Canadians will be affected each year by cancer. The good news is that actually our treatments are getting better and cancer mortality in Canada is actually going down. So cancer death rates are decreasing for both males and females over the last couple of decades. And these are cancer statistics from the Canadian Cancer Society. So if anyone is online from CCS, thank you. And I think a lot of this progress in the last couple decades has really been due to our greater understanding of the human genome, not only in sort of the normal cells, the normal genome, and human development, but also in cancer, which we call the cancer genome. And so again, I'm just going to give you a little primer on genomics before launching into our guest speakers. So the human genome, what is it? So in my mind, the human genome is basically like a library of life, but it's written in the code of DNA. And so this library contains books, and all those books contain all the instructions that you need, or the cell needs, to make all the parts that it needs to make a functional unit. So if we were talking about cars, for example, this would contain all the instructions to make all the parts, the brakes, even the little screws, the steering wheel, and also the instructions to assemble that into a functional unit, the car. But of course, we're not talking about cars. We're talking about cells, and in your cells, these books are what we call chromosomes, and we would have 48 chromosomes instead of the 20 books shown here, and those package all your DNA, right? And that DNA, those books are divided into chapters and sections, and those chapters and sections are genes, so those are the instructions on how to make the parts of the cell. And those parts of the cell are things like proteins, and then those proteins are assembled, packaged into a cell. And of course, it includes all the instructions to assemble cells into organs, and organs into the tissues, and the tissues into the whole human. So this is a very complex system, and of course, things can go wrong. We can have errors in the DNA. So one kind of alteration is that if you have mistakes in the instructions, maybe you don't make the parts right? Maybe your brakes don't, turn out right. And then instead of a car, you end up with, the proverbial lemon that doesn't work. But in our DNA and in cancer, instead, if we have a mutation in genes, we can affect the function of the proteins of the cell. And then that can cause the cell to proliferate too much. But really, it's a little bit more complicated than that, because it doesn't really require one type of alteration. There's not just one mistake and then everything goes, turns into cancer. There are many different kinds of errors that can happen in the genome. So again, anywhere in this vast library of information about how your cell should behave and what they should become. Some of those, which you'll hear later on tonight, you'll hear about mutations, and those might be little errors. Let's say you have an instruction and you're missing the word not. That might be a very important mutation. Or let's say it changes the meaning of a word in a sentence. That would be a mutation. A little section is sort of has a little mistake in it, like a typo. And again, those particular types of typos can cause things to malfunction. The brakes don't work, or your gas pedal is set at a certain speed, or your cruise control is set at a certain speed. So those can cause malfunction of the cell. But those aren't the only kind of alterations in the genome. So you'll also hear about cancer genomic rearrangements, so rearrangements in the DNA. And that'd be if you have, like, let's say you start out reading one book or reading one recipe. You might actually have a whole other piece of DNA stuck on. So you start out with Moby Dick and you end up with who knows what. So those rearrangements can also cause problems, and they can sometimes cause you to produce things like fusion proteins that are half of one protein and half of another protein. And of course, those things don't function very well either. And then another class of errors that you can have in the genome are copy number alterations. So in this kind of mistake in your genome, there can be many, many copies, let's say, of instructions to make a particular protein. And if that protein is something that's responsible for giving you a growth signal, please cell grow, proliferate, you could have abnormal proliferation of cells and eventually lead to cancer. So let's say you have too many motors in your car. Well, that's going to drive your car a little bit, and probably not in a good way. So after kind of going through all these different kinds of mutations or alterations, I'll also say it takes more than one alteration to create a cancer. So often you'll need something like too much gas and losing the brakes for your car to go out of control. So here, cancer is actually an aggregate of several different mutations within the same cells that all sort of cumulatively lead to dysregulated growth. So in the past sessions, in the couple of other sessions we've had in Late Night Lab, we talked about the burden of cancer in the population. So at a population level, what are risks? How frequent is it? We've talked about some of the overview of different kinds of therapies, including, how maybe immune cells might interact with cancer cells, and, this concept of DNA. We've talked about cancer in different special populations, like cancer in childhood. But tonight, we're going to focus on two particular questions. Is, with all this DNA damage I've talked about, the leading to dysregulated growth control and cancer, how does our DNA become damaged in the first place? So that's one area that you're going to hear about. And then the second area that you're going to hear about is how can that knowledge about DNA damage and the mutations and alterations that these cancers carry, how can that be used to improve therapy for our patients today? And then how might that be used in the future? So just with that brief introduction, I'm going to stop sharing my screen and turn it over to, oh, actually not quite yet, turn it over to Dr. Erin Gedarzi. Erin, are you able to share?
Okay. Thank you very much, Jennifer. So hopefully everybody can see that. So I'm going to begin my journey, which is to tell you about DNA damage, DNA repair, with a discussion of why across the world our burden of cancer actually is quite different. And if you look at the #1 most diagnosed cancer types across the earth, it is very different. Here in North America, it is lung cancer. That is the #1 diagnosis, where, for example, in Southern Europe, we have colorectal cancer being the #1 most diagnosed. In Central Africa, it is liver cancer. In South America, it is breast. And in South Asia, we actually have cervical cancer. So why is this the case? It is not equivalent across the earth. And not only is it not equivalent across where we live, but it also is not equivalent by our age. So if we just consider lung cancer, the #1 most diagnosed cancer type here in North America, there's a huge difference. And while it is true, like many cancers, lung cancer is overwhelmingly diagnosed in the aged, those 60 and over, there's a significant proportion, up to a fifth, who are actually considered young, with many of those and indeed, just under 700 Canadians last year between the ages of 15 to 49. So what's going on and why is that the case? Well, it's all about our DNA. And Jennifer nicely introduced the concept of chromosomes, which are the units that you'll find our DNA package in every single cell of your entire body. And what I'm showing you here in the left-hand side of your screen is a nice, normal, in this case, a girl cell. You can see there it's got 2 little X chromosomes. That's the signature of the female sex. And there's 2 of everything else. And this is how we're born. We have 1 set of chromosomes from mother and the other set from father. And hopefully, as we're born, these are all nice, intact, and undamaged. However, as we go through life, they are subject to a variety of exposures as well as fundamental processes that damage them. And like cutting a piece of rope, a chromosome can actually be sheared in 2. You can also fray the rope, you can snip at it, you can paint it with something, you can drop some acid and eat a little bit of it away, figuratively speaking, to damage that DNA. And in fact, many of the things that we encounter throughout life will do this. This not only contributes to our aging fundamentally, but also our risk of diseases such as cancer. And what you're seeing here is a few examples of the things that can damage our DNA. These include things like tobacco use, air pollution, chronic inflammation, and radiation sources such as ultraviolet sunlight and ionizing radiation, which is in fact my expertise, and we'll be hearing a little bit more about later. But to illustrate an example, let's just consider sunlight. If you're in Calgary right now, it's a nice sunny afternoon. And if you're going outside with that sunscreen, this is what's going to be happening to the DNA of your skin. And in fact, on nice sunny days, you will be encountering ultraviolet A and B sunlight. Depending where you are in the world, ultraviolet C may or may not get through because that is usually filtered out by the ozone layer. However, for example, if you're in Australia, some of that might actually get through. What those ultraviolet rays are doing is actually fundamentally chemically altering our DNA everywhere it hits. It actually kinks the DNA. That not only is very toxic to the cells, in fact, it's the #1 reason why we actually can get a sunburn, because if enough of that DNA damage accumulates, you will actually kill the cells. We all perhaps had a sunburn in our life. That's what's happening to those cells. But if the cell just gets enough damage and doesn't die, which is often the case, then you will cause mutations which can drive cancer. Now, thankfully, we all have repair mechanisms that work to resolve this as fast as it can and as accurately as it can. So, for example, if you're exposed to an X-ray radiation, that might go through the DNA of our cells. And here again, if you look at that diagram I showed you earlier, we're expanding that out and looking at the little chromosomes. So here you're seeing 5 little cartoon chromosomes. And every single one of those little green things represents where that X-ray might have hit and caused a break in the DNA. Now, thankfully, our cells are really good at rejoining that type of damage, which is why, you know, regular low-dose medical X-ray radiation is extremely safe and very beneficial in terms of telling you what's wrong with you if you, for example, break a rib or the dentist needs to see your teeth. But the damage repair mechanisms we have actually do depend on the type of damage they're encountering. And for example, there are other types of damage that are much more challenging for our bodies to deal with. And in fact, the more challenging, the more likely it is to make an error. And here you're seeing one example of a type of DNA damage repair error that can lead to a mutation. So here you're seeing 2 chromosomes, the purple one and the pink one. For example, that might be exposed to a type of radiation, get broken, but the cell rejoins them, but it gets it wrong. It puts the pink with the purple and the purple with the pink, and that can really start driving cancer mutations if enough of those happen in the right place at the right time, or perhaps arguably the wrong place at the wrong time. Certain types of damage are much more likely to do this than others. One example for that is particle radiation. Here you're seeing an example of 2 cells. On the left, in A is a nice, normal, in this case, boy cell. It has X and a Y chromosome, 2 of everything else. And in B, you're seeing the typical genetic fingerprint of cancer. You can see how some of these chromosomes are just missing altogether. Number 13 is missing entirely. Others have more than 2 copies, up to 5, 6 copies. And at the bottom, there are these monsters we can't even recognize as coming from mother and father anymore. They've been broken and rejoined incorrectly so many times we can't recognize them. Particle radiation, for example, is very good at doing this. Our cells encounter many different types of radiation through their life. The X-ray, very simple, as I say, not particularly dangerous for you, goes through us very quickly. That's photon radiation. Particles, on the other hand, go through us very, very slowly, depositing a lot of energy. The analogy here, it's like shooting a single bullet at the side of a building versus for example, multiple square kilometer carpet bombing of the building. Very different in terms of quality. And when your cells encounter this kind of DNA damage, which they often do in the form of radon gas in our houses. Radon emits particle radiation. We can't sense it because human senses can't tell it's there, but our bodies know it's there because the radon releases particle radiation that move at half the speed of light with enough energy to dent bulletproof plastic. Well, when your cell encounters that, multiple things will happen. The particle will go through. It's not widely scattered anymore. It's very dense. And the DNA in that area where it hits actually gets shredded. It's not just a question of rejoining 2 little pieces of rope anymore. Rather, you have a complete shredding of the area, and those little pieces will get lost. Not only that, you've got multiple pieces, multiple chromosomes all broken in the same area. And fundamentally, our cells are just not good at dealing with that type of damage, driving them from the normal to the cancerous much more quickly and at lower doses compared to, say, a photon. So the type of DNA damage does matter. Now, the International Agency for Research on Cancer, IARC, you can see the link there if you want more information about them, actually categorizes all the things that we know as humans will give us cancer. They classify it in groups 1 through 4, and they just indicate how well we know they cause cancer. So group 1 is not necessarily more dangerous than 2A. Group 1 classifies those compounds in our lives that we absolutely know cause cancer in both humans and animals. 2A is probably, 2B possibly, and so on down. So in terms of what's in group 1, things that we absolutely know cause cancer, here you're seeing some of those in the top 10. Things like alcohol. That's in fact the #1 way in this part of the world that we are exposed to cancer-causing compounds. Hydrocarbons from air pollution, ultraviolet sunlight I spoke about, radon gas I spoke about, and you can see some other examples there. These collectively we will encounter throughout our life, and they make up what is called our exposome. Just like our genetics make up our genome, these exposures make up our exposome. And they will be different for every single one of us, depending on our diet, our medication, our health, the infectious disease we encounter throughout life, and the pollutants, whether we do or don't encounter them and how much, make up our exposome. And this matters for your health. Each of us has a very individualized exposome. Here is a figure that I borrowed from Public Health Ontario for the annual cases caused by each of the many types of cancer-causing agents that they have out there. And you can see solar radiation, radon gas, air pollution, and things like that are high on the list out there. It's very similar here in the west of Canada. And these each make up our personal exposome, the relative contribution of each of those to us. Now, these can be modified by a variety of things, as you might imagine. They depend on your occupation, they depend on your behavior, they depend on the built environment, your diet, and not only that, your location, not here in the city, but also across the earth. So let's break those down a few. So cancer risk can actually be strongly modified by your occupation. There's a variety of examples of this, and I'll use UV sunlight once more. If you look at by occupation, data from Carcinogen Exposure Canada, CAREX Canada, indicates that our construction workers and our farmers are by far and away the most exposed to ultraviolet sunlight. That makes sense. They're outside all day. Where, for example, if you're in an office building, you're much less likely to have ultraviolet sunlight as your major exposure, but perhaps radon gas would be more a problem for you. Now, behavior also matters. Alcohol, we spoke about alcohol. Well, as you might imagine, that's also a function of age and gender. If you're young and male, you're much more likely to be exposed to alcohol because of your culture that you live in, at least in this part of the planet. And so your behavior matters, and that is true for many, many, many different types of cancer-causing agents. Not only is our behavior and our job matter, but where we're living and where we're working and going to school matters. Our built environment, the architecture, the engineering of where we live can actually concentrate some of these cancer-causing compounds or dilute them. And one example of that is radon gas. Normally in your yard or in a field, radon is coming out of the ground and diluting to nothing all the time. But we've built, at least in this part of the world, houses that are increasingly capturing, containing, and concentrating that to extremely dangerous levels. And that is a function of our built environment. In fact, radon, by and large, is a man-made problem because of this region, our built environment. As a shout out to some of the projects that I actually run at the Charbonneau Cancer Institute, we lead Evict Radon, which is a national study aimed at understanding and trying to engineer out radon. Why is this important? Well, if you actually look at radon gas in the Canadian built environment, it's gotten progressively worse since the end of World War II. You can contrast that to what our cousins across the Atlantic in Sweden have done, where between the 1950s and 70s to today, although we both started off with approximately the same amount of radon in our houses, they've successfully engineered theirs out, where ours has gone up and up and up and shows no sign of declining. We're trying to understand exactly why that is and reach out. So if you want to learn more about that, you can go to evictradon.org. But it does say that where you live, the age of your house, the type of your house, the newness of your house makes a difference. And that's true for radon, but that's true for many other things as well. Your diet makes a big difference. We encounter many of these things that cause damage to our DNA. Often that occurs through highly toxic reactive oxygen species. Well, depending on the amount of antioxidants in your diet, that can counteract that and actually reduce your risk, at least within a certain window. And so things like fresh fruit, certain berries, dark chocolate, I'm sure you'd be glad to hear, nuts, and others are high in antioxidants and can be beneficial, at least within certain realms there. And then finally, as I said before, they differ globally. And that's because, for example, many of these exposures do differ globally. Alcohol consumption differs very widely. In many parts of the Middle East, it's not allowed to drink alcohol, so that, of course, the exposure to that is much less. Other parts of the world are very high in air pollution. Here in North America, depending on where you live, you may have more or less radon exposure. So it depends where you live, your actual exposures. Now, the final two things that modify our risk to cancer are our age and our genetics. And so, for example, our children are fundamentally at risk. All children are hypersensitive to cancer-causing agents like this. This is because they're still developing. Growing organs are fundamentally at risk if they encounter DNA damage as they're growing, because that damage will be propagated through every subsequent piece of growing tissue. They weigh less than we do as adults, right? So, for example, they get 100 units of a certain amount of a cancer-causing compound, same as we would, but because we weigh 3 times larger than they do, they actually get 3 times more exposed overall in terms of concentration. They also breathe faster. Many things we encounter that give us cancer, we actually breathe them in. Radon, for example, air pollution, asbestos. Little kids breathe faster. If you're carrying a little baby in your arms, he or she is most likely breathing about 3 times as fast as you are because of little lungs. And the most brutal truth is our kids have the most life left. Exposure at the youngest ages, where you may have hopefully 75, 80 years left in you, matters, because now 80 years is a long time from that exposure at, say, age 5, to go without developing a cancer. Where, for example, if you are 75 when you're first exposed, you're most likely not going to succumb to a cancer because of the latency period. Cancers from these often take between 5 to 30 years, depending on the type and the dose that you're exposed to develop. Now, some adults remain sensitive, and this is where my final point, genetics, really comes into it. So we know our kids are all sensitive, but most of us grow out of it. But some adults remain sensitive, and this really does matter for the genetics. For example, what I'm showing you here is the induction of DNA damage in a normal skin cell. So the DNA of the cell in this case is blue. You can see the little blue round things. And within them, little green dots. Those little green dots all represent a break in the DNA. And very quickly, within half an hour, you can see there's lots there after the damage is induced. But within about a day, most of them are gone. And certainly by 3 days, they're all gone. We're actually really good at repairing those. Most of us are anyway. Some of us, however, as I say, as adults remain sensitive. And there are those amongst us who have a reduced capacity to carry out effective and accurate DNA repair. How do we know this? Well, we see this all the time in our clinics at Tom Baker Cancer Center. For example, those going for radiation therapy. The underlying fact with this is thought to be the single nucleotide polymorphism. It's a big long name, but it's actually pretty simple. It simply means variations between us. In my DNA sequence, maybe I have like this, CAG, but perhaps you have a CGC. That single difference, the A to the G, matters depending on where it is. Sometimes it's completely benign. Sometimes it just gives me brown eyes and you blue eyes. Sometimes it changes the color of our hair, but sometimes it changes our ability to repair DNA. And as I say, we can see this in our clinics. For example, 1 in 30 of the patients that we encounter who receive radiation therapy for a cancer type will over-respond to that radiation. That over-response can range from a little bit of a rash you wouldn't have expected to something even much more severe like long-term disability or even death. And there's no way for us to know in advance who is who before it goes in there. But this speaks to the undiagnosed susceptibility to radiation, but also many cancers prevalent amongst us because of these little variations that are prevalent throughout. This, for example, in radiation sensitivity corresponds to 1.13 million Canadians. This matters because, of course, we don't just receive radiation in terms of our medical treatment, but, for example, maybe your house has radon gas in it. Well, that's also radiation. And are you sensitive or are you not? Well, that's important information to find out. So it is all about our DNA, and it matters what our genetics are, and it matters what our exposures are that modify our DNA. Now, one of those two things we can control, and this is where I'll end, to say that in terms of our exposome, we do have a degree of control over this. We also, if you have children, you have a degree of control over what your children's exposures are. We can put sunscreen on. We can test ours for radon. If it's high, mitigate it. We can avoid certain other things, tobacco smoke, if possible. I know it's a chemical addiction, but these are things that we can actually take control over with our behavior. So I'll end by saying to protect our DNA, we have to know how it is damaged and know those exposures. And I'll do a shout out to the Canada Research Chair Program. I am the Canada Research Chair for Radiation Exposure Disease. If you want to know more, you can Google search me. I'm the only one out there. And if you want to know more about radon or potentially test your house with cancer researchers, go to evictradon.org. Thank you very much. And I will give it over to Dr. Gwyn Bebb. Gwyn, over to you.
Thank you, Aaron. Good evening, everybody. Even though I can't see you all, it's an absolute pleasure to be part of this, and it's an honor to be invited to share some knowledge and experience about this very important topic. We're going to try a more advanced piece of technology here by having Dr. Chan, I believe, share the screen with me. Is that right, Jennifer?
Yes. Sorry, I'm gonna. Did you hit the green bubble, or did you want me to run your slides?
I don't see my slides at the moment at all. So do you want to share your screen and then I can advance? Is that possible?
Oh yeah, yes. Actually, I'm going to do that right now. Yeah.
As way of introduction, I'm a medical oncologist at Tom Baker. I have an academic position at the university. But I'm here to share with you my experience and outlook on my life as a medical oncologist who treats lung cancer. And what I'd like to do tonight is follow on very, very smoothly, if I can, from what Dr. Chan and Dr. Gudarzi have just introduced us to. That is the nature by which changes in our genome, in our DNA, can bring about cancer, and how an increased understanding of that process can lead to improvements in our treatments. I want to touch on three things. I want to touch on how point mutations, or those small little changes in DNA that Aaron mentioned, can bring about cancer, and how we can target those to change disease, how those rearrangements that we were talking about can do the same, and then how a multitude of mutations that cause a heavy tumor mutation burden can affect a cell in a way that leads to cancer as well. Interwoven with all those themes, I want to introduce you to some of the work that's going on here in Calgary at the university and at the Charbonneau in particular, and share with you a little bit of optimism that we have about how things are progressing, and yet at the same time the kind of disappointment that we still don't quite have the magic wand that changes the experience of our patients to the degree that we would like. So tomorrow morning, I have a clinic at Tom Baker. I will see 3 new patients. And to each of those, I will have to introduce the fact that they have an incurable disease. That this disease that we're trying to treat is going to take their life. And this depressing fact reflects the very high mortality that we have with lung cancer. And one of the questions we often get here, why is the mortality so high? And if you look at this pie chart here, what we'll see is clearly the majority of our patients present with advanced disease, stage 4 and stage 3. So, close to 2/3 of our patients have very challenging disease, and certainly with stage 4, we do not cure anybody. So there's many reasons for this, but the fact that lung cancer can lurk and hide and not cause any symptoms is probably a big part of the late presentation, and the fact that we're late in the game in this setting. Now, if we look at lung cancer, although it has a common thread in its progression, and that it kills more cancers than any of the other cancers combined, in fact, on detailed study, and particularly over the last 15 years, we've got to grips with the fact that not all lung cancers are the same. There is a range of mutations that we can detect in patients' lung cancer samples. And many of these, over the last 15 years, a period of time that I'm almost calling a revolution. If it's not a revolution, it's a massive change, because an understanding of these mutations and the role they play in cancer has led us to develop new treatments that target these mutations and change the clinical course of the disease completely. You see there in the top part of the table there, the EGFR and ALK mutations. Those are the ones I want to talk about. The EGFR is a kind of a very specific mutation in a, we go back there, sorry.
Sorry.
Okay. The ALK represents a rearrangement. And now we have treatments also for something called ROS1 and BRAF. And if you look at the table below, that list there, HER2, KRAS, MEK1, MET, NTRK, RET. At the recent conference held virtually at ASCO, we learned of new therapies coming down the road for each of these. Now, one of the interesting things I want you to grasp here is that as the list goes down there, the frequency or the prevalence of these mutations tends to decrease. And so one of the challenges is identifying these mutations and conducting clinical trials that have enough patients in them to tell us that our new treatments are actually effective. So let's go to the EGFR mutation. This is something that we were just getting to grips with when I came to Calgary about 15 years ago. And in about 10 to 35% of our non-small cell lung cancer patients, we can identify an EGFR mutation, a small change in the gene coding for this protein called EGFR. Our data here in Alberta suggests that probably 17% of our patients have this in their cancer. But if you go to some of the denser populated areas in Southeast Asia, in Taiwan, China, Hong Kong, and Japan, you'll find the prevalence as high in some case series as 65 to 70%. Now, we look at this EGFR mutation in a little bit more detail. What we find, in fact, is that these changes in the DNA.
Do you see anything there that says remote control?
In the DNA has a profound effect on the structure of the EGFR molecule that you see on the left of the chart there. And I want to draw your attention to particularly what you see there as exon 19 and then this locus here, because those are the sites where we find a lot of mutation. If you look at the pie chart on the right, the blue piece on the right of the pie and the gray bit in the bottom left illustrates the commonest mutations we find there, representing about 3/4 of our patients with EGFR mutations. And those are what we call the activating mutations in EGFR. And they have two consequences that are important. One is they activate the molecule in such a way that it can drive the cancer almost on its own. And the second consequence of this mutation is that it renders the molecule, the structure of the EGFR, particularly susceptible to inhibition by a new class of drugs, which are called EGFR tyrosine kinase inhibitors. Let's go to the next slide.
So, yeah, thank you, Gwen. Just let me know when you need to advance.
And what you see before you here is a CT scan through the upper chest, probably across here, of a patient of mine. We're looking up the chest, and the CT scan on the left shows you a huge burden of disease. What we should see there is a lot of black, representing the air that's filling the lungs, but instead what you see is a lot of white substance, and that represents a very, very nasty mixture of cancer, inflammation, and indeed some infection. You look at the CT scan on the right of the same patient 3 months later, and what you see there is those areas that were previously white are now filled with air, and there's almost complete resolution of the cancer and the consequent infections and inflammation arranged with it. You can see traces of it, but this is a remarkable transformation. The patients who experience this kind of response go from being almost bedbound to leading a completely normal life. And the other great thing about this new class of drugs is the fact that they have a very, very tolerable side effect profile. So if we're keeping patients on these for many months, this is an absolute requirement, something that we would not normally be able to do with standard chemotherapy. Let's go to the next slide. The other exciting thing about this arena right now is that, as you can see from this, what we call survival curve, this is a number of patients who still have stable disease over a period of time from the initiation of treatment. These are patients who, after having responded to that kind of drug that I showed you in the previous CT scan, have relapsed and are now being treated in the second-line setting. And these are patients who have a second kind of mutation in the EGFR gene, a specific resistance mutation called T1090M. And when this mutation arises, the sensitivity of the cancer to the original treatment disappears. But what this graph shows you is that our third generation tyrosine kinase inhibitor, this third generation drug that targets the EGFR mutated protein, osimertinib, has a significant impact on these resistant tumors. So I think what has developed in the last 15 years is, first of all, an understanding of the role of these mutations and our ability to target them, and then the development of second and third generation drugs that do a better job each time. Let's go to the next slide. Now, one of the things that is very interesting in this field is that as the frequency or prevalence of these mutations becomes small in our population, our ability to understand what's going on and conduct clinical trials is handicapped because it is more and more difficult to get enough patients to numerically power the trial to show statistically significant difference. And one of the things we've helped address here is a collection of what we call real-world evidence. This is through what we call the Glands Look Lung Cancer Database, a database of lung cancer patients in Calgary that looks at outcomes, treatments, responses, and to some extent quality of life in lung cancer patients over the last 15 years. And it's been called Glands Look because the funds that started and subsequently keep it going were initially raised by John Glands, who organized a series of golf courses, and Jim Look, who persuaded two of his friends in the oil and gas sector to come and support it. Now, what kind of use can we get from this? That's great. Go to the next one, Jen, please. Yeah. So let's look at the ALK rearrangements. We just looked at EGFR mutations. Now we want to look at ALK rearrangements. And this is the second big change that we've seen in lung cancer, the discovery of the role of this rearrangement in driving cancers forward. It is less common than EGFR, probably present in 3 to 7% of our population. Here in Canada, we think is between 2.5 and 3.5% of the population. A series of drugs have been developed that target this. We're on to the fourth generation in this class of drugs. And here you see not a CT scan this time, but a combination of a PET and a CT scan. A PET scan, as many of you will have heard, is a scan that picks up metabolically active or glucose-utilizing tissue in the body. And yes, it does pick out the brain, you can see at the top there. But if you look at the figure on the left there is a lot of bright yellow areas, particularly in the midriff there, around by the liver, in the lung, and in the abdomen. And that actually is the extent of the cancer burden in this patient with lung cancer, which happens to have the ALK translocation. A 14-day treatment with an ALK inhibitor gets you the PET scan that you see on the right there. And the difference is absolutely staggering. And I think, before this understanding, we never witnessed these kind of responses and changes in patients, where it was nicknamed the Lazarus response, where somebody, again, bedbound, becomes active and almost normal within a 2-week period when they're treated with a low toxicity targeted agent. So this has profoundly changed our experience. Let's go to the next slide. What have we found to be the case in Alberta? Well, we've just been requested by a combination of Health Canada and Pfizer to look at the experience of ALK-positive lung cancer patients in Calgary. And this is what you see here as a survival curve where the median overall survival, that is the time to when half our patients have died, is 46.6 months. That's almost 4 years. That is quite a staggering statistic. And it's something that 15 years ago would have been incomprehensible to us. And this kind of data derived from a database such as the Glands Look helps verify the fact that these agents, as well as showing benefits in the clinical trials in which they're tested, also show benefits in the real-world setting to real patients in our clinics. Of course, what is a little bit disappointing about all this is the fact that none of these treatments are curative. And for reasons that we still don't completely understand, despite the onset of second, third, fourth generation drugs for these EGFR mutations and ALK rearrangements, we still do not cure our patients. And ultimately, the cancer evolves in such a way that we can no longer treat it. And yet, we have still seen a revolution in patient experience in general over this time period. Now, what you see in front of you is very similar to what Gorazi showed a little bit earlier. That is the kind of grotesque chromosomal appearance of a cancer cell. And this is probably what we see more commonly in lung cancer. Instead of having single driver mutations or a single driver rearrangement, we have this smorgasbord of mutations. And trying to target each of these is clearly very, very challenging. Now, one of the interesting ramifications of this mutational smorgasbord or heavy mutation burden is in fact illustrated in the next slide. Remember that each of these genes codes for a protein, and the more aberrant your genome, the more changes and mutations there are, the more likely it is that you will have structurally different proteins that may have therapeutic implications, as I'll allude to in a couple of slides. This is a very interesting slide. It's now a kind of iconic picture. If you look from left to right there, you'll see a series of tumor types from Ewing sarcoma all the way to melanoma at the right there. And just left of melanoma, you'll see two kinds of lung cancers, the adenocarcinomas and the squamous cell carcinomas. From bottom to top, numerically, you get an increase in the number of mutations seen per cancer cell. So on the left of the chart, you'll see tumors with a relatively low, what we call mutation burden. But over to the right is a series of tumors that have high mutation burden, and therein lies many solid tumors, including lung cancer. Now, the other thing to say about that, actually, if you look at the line that's associated with lung adenocarcinoma there, it's actually quite a wide spread. So there are some adenocarcinomas of the lung that have a very low tumor mutation burden, and those are probably the ones with the driver mutations that we spoke about just now, the EGFR and alpha-positive patients. But up at the top of that line, you'll see a lot of tumors with a heavy mutation burden. So what does this have in terms of implications for therapy? Well, I think 2 years ago, the Nobel Prize for Medicine was awarded to these two fantastic scientists and researchers, Dr. Honjo from Japan and Dr. Allison from the United States. And what they had worked on for their career, essentially, was a greater understanding of the immune relationship between a cancer and the patient's normal immune system. We've always been puzzled as to why cancers do not respond, or why don't they trigger the immune system in such a way that the immune system can get rid of them. And what these two scientists have identified is that there are some blockages in the body that can prevent the immune system from reacting to a cancer in a way that makes it go away. And what has evolved from that discovery are treatments that undo that blockade to allow the immune system to resume its attack on a cancer. And the cancers that probably do best are the ones that are going to have the mutation burden that we spoke about earlier. They're going to have so many aberrant, abnormal proteins that they will be recognized as non-self and therefore are more likely to trigger an immune response. Let's go to the next slide, Jen. This shows you a CT scan from another patient of mine who was treated with immunotherapy. And there on the left, you can see in August of 2017, a very large, nasty mass, as well as metastases in other parts of the body. But by December 2018, this has significantly shrunk. Now, and then let's go to the next one. There's a slightly different view of the same tumor looking, instead of looking up the body, we're looking at it from the front. There's the tumor again, and it's shrunk quite magnificently. And so what we see here is, instead of the kind of Lazarus responses that we saw with the other driver mutation positive cancers, whereby we got huge shrinkage and Lazarus kind of rise from the dead responses within a week or 2 months. Here we see a more gradual attack on the cancer by the immune system, by those patients whose tumors respond to this treatment. Now, this again, I think, represents the second big revolution that I've witnessed in the last 15 years in lung cancer. It is quite phenomenal to see it in action. And despite the slow responses that we see on CT scan, I think our patients do feel better much more quickly than that. Disappointingly, though, it is still difficult to say that this is curative. We do have patients who do well on this for 2, 3, even 4 years. But to describe it as truly curative is very difficult right now. And overall, despite these advances and a better understanding of the immune system, we still get responses only in about 30% of our patients. Let's go to the next slide. Keep going to get the three things. So one additional aspect that is kind of become quite a hot topic in the last 2 to 3 years here is the human microbiome. So we focus right now on trying to understand the genome of the cancer and how those mutations drive the cancer and how they can maybe be used to elicit an immune response. But there's been a series of observations made that our responses to these immunotherapies actually depend on the nature of the microbiome, particularly the microbiome in our bowels. And this chart shows you how many microbes actually coexist with us. In some ways, as soon as you look at the fact that our human genes of 23,000 or so make up a tiny fraction of the genes that actually exist in our body, is actually quite humbling. What does actually constitute me or you? Anyway, that's a philosophical question. But nonetheless, I think this is going to be an aspect of significant research over the next 3 years, is trying to understand how the microbiome influences our response to immunotherapy. And once we understand it, how can we modulate the microbiome to optimize treatment responses in the immunotherapy era? And then to the last slide, I happen also to be a leader of this initiative. It's called Precision Oncology and Experimental Therapeutics. And one of our key drivers is to create an ensemble of researchers across the University of Calgary, all focused on trying to better understand and deliver precision oncology. And one of the key attributes of our program is the fact that we're collecting stool and saliva and urine samples as well as blood samples on our patients following their journey through their cancer treatment. And hopefully from this, we can utilize some of the excellent capability that we have at the University of Calgary, including our world-leading microbiome center, to better understand how mutations, not just mutations in our tumors, but mutations and the genomic makeup of the bugs that coexist with us can influence our response to treatment. So thank you very much for your attention. I'm very happy to join my colleagues here to answer questions.
Okay. Yes, thanks, Gwen, for that. And sorry that I was unable to give you control of your own slides.
Okay.
I think it went okay. Aaron, do you want to turn your video on? So we will have our question and answer session now. And I'm going to look at the chat, so you can feel free to use the chat. We did get some questions beforehand, so I'll just start with you guys. And sorry I didn't have the introductions. Aaron Godarzi obviously is one of our DNA damage and repair scientists at the Charbonneau Cancer Institute, and again, Canada Research Chair in DNA damage. And Gwen Bev is a medical oncologist specializing in lung cancer, but treats other cancers as well at the Tom Baker Cancer Center, and is also a clinician researcher. So let me just get started. I'll kind of just I'll put the question out there. Sometimes I'll direct it at you. You can't see me pointing at you, Aaron. You're on the right of my screen. Or you, Gwen, on the left of my screen. But one of the questions that we had gotten from a few people was around the idea of the novel therapies and access to novel therapies, because they are, as you say, showing to be less toxic. They are showing, at least in some instances, to be very effective. There are questions about how do you access those, in Canada, in trials. Are there navigators? Can you give some kind of guidance there?
Yeah, that's an excellent question, and it's a source of some angst very often. I think there are a couple of challenges that we should address separately. The first is, how can we generate evidence to justify the use of these agents? So there's a lot of promising things in phase 1 trials, a lot of promising things in the lab, but drugs are approved after they've demonstrated efficacy in a bigger clinical trial, usually a phase 3 trial with large numbers, where the new treatment is compared and shown to be better than our existing treatments. as I alluded to previously, this is becoming a little bit more of a challenge in those subgroups of patients where we have very few numbers to be able to put into a clinical trial. So that's one challenge. I think a second challenge is once a drug has demonstrated benefit in a phase 3 clinical trial, the next challenge is getting access to it. And there's no doubt that in the United States, the federal drug, the FDA is very keen on approving things, but the FDA has the advantage of not having to cover those expenses. Health Canada is a little bit more cautious, but usually follows suit within 4 to 6 weeks. But the biggest challenge within Canada after that is getting funding. And so there lies a challenge for the health minister of each province to find a budget to cover the cost of these drugs. And the cost of these drugs is quite profound. For our immunological therapies, we're talking about at least $100,000 a year per patient. And for some of these newer oral pills, we're talking 10 grand a month as well. So these are not simple questions to answer. As you rightly pointed out, one way of trying to get access is to be involved in clinical trials. There is a website at the TBCC that lists the clinical trials that are ongoing, but it is obviously a small number of clinical trials compared to the breadth of new drugs that are being tested all over the world. So I don't have a perfect answer for you there, but advocacy is important in trying to drive this agenda forward, and probably we need to do a better job of hosting more clinical trials, and somehow we need to find a way of forcing some of our pharma partners who invest a lot of money in the development of these drugs. We have to somehow partner with them to reduce the cost of these to our healthcare authorities.
Thanks, Gwyn. There's a question here. I think it would be up your alley, Aaron. Is it possible to quantitate or quantify in some way the exposome? And you might be doing that. Or
yeah, so this is a very exciting area of ongoing cancer prevention and early diagnosis research right now. So what we can do for many exposures is quantify an individual exposure. So for example, if you go out and test your house right now for radon and continue testing every property that you might live in for the rest of your life, you'll largely know your radon exposure history. So that component of your exposome you will know. Similarly, we've been doing that for asbestos and tobacco use for many years. One of the big, bold new areas that researchers are looking at now are to understand mixed exposures of everything that you might have been exposed to going back to childhood. Largely, the clues to many of these will be in our bodies. We just have to find out what the correct biological markers or biomarkers are for those. And so that's a very active area of research. So my answer is yes, you can for many things individually right now. And many people are working towards a sort of more comprehensive measurement of the exposome for everybody.
Thanks, Aaron. One question that I think you touched upon, Aaron, in your talk. We've been talking about acquired DNA damage. But you had a little bit in there about susceptibility as well. And we've talked about sort of targeting the acquired damage or targeting these things that we've picked up. Are there ways to target the inherited mutations? Do they also give opportunities for treatment for cancer?
So inherited mutations or inherited susceptibilities is actually really important to understand. I give the example of the 1 in 30 people that we encounter who over-respond to radiation. So that's an example of understanding people's radiation sensitivity before you might commence any kind of treatment. And yes, the technology to understand if you are radiation sensitive before you would undergo treatment does exist. It's typically used in the context of childhood bone marrow transplant. Now, those differences amongst us that we can understand comes with genetic sequencing, and there are many opportunities out there right now, both publicly and privately, for you to get your genome sequenced. Largely things like 23andMe and others, they're only looking for things that'll tell you about your ancestry. Those are not necessarily or at all medically informative, and there's a lot of privacy questions surrounding those. So I say we do have the technology to do that kind of thing now, and in many cases it is being applied where the most medical good can be done. But it is something that is rapidly evolving and one should be cautious about.
Gwyn, do you have anything also to add to that?
Yeah, I think what should we say, the characteristics of our cancer are often very difficult to assess, and when we're giving treatment, what we're trying to do is target the cancer while sparing the body. So on the one hand, we want to try to assess the body sensitivity to an agent, but we also want to assess the tumor sensitivity to the agent. I think it's a very complex area to try to kind of build into general clinical practice. The other thing I would say is that a lot of our genomic profiling has revealed some very useful targetable defects in probably 5 to 10% of our patients' cancers, but there is a vast majority so far whose treatment has not been usefully affected by these exercises. And so we still struggle to understand exactly how to utilize the information that we glean from genetic profiling and genetic sequencing. And clearly, I think what we're learning now is that to be really precise in our oncology strategies, we need to invoke an understanding of things like the microbiome, things like the immunome, and other things related to the body that all interact in this very complex way that leads to cancer and its eradication.
I guess the one example I was thinking of might be like the familial breast and ovarian cancers with PARP inhibitors. So there are things that we're doing that are actually DNA damage, I guess, related genes, but that could offer part of those therapies as well. There was I forgot whoever said this, but there are privacy issues. That was one of our questions as well. We're talking about DNA, and you can identify people from their DNA. Is there a worry now that we're moving into the molecular era of testing about patient privacy and identification? I guess maybe on the clinical side first, and then Aaron, you can chime in.
Yes, there is a worry. When I first came here 15 years ago, we used to say in our ethics application that all data would be carefully stored in a safe with a lock in a room that was also locked. That clearly isn't the case anymore. Everything's stored on servers, in databases, and we know that banks are always worried about people hacking into their systems, and so the same is true for research databases. We know that the Alberta Health Services and the University of Calgary have developed pretty high security technology to help secure the safekeeping of such data, but nothing at the moment is insurmountable. So this is a significant risk, I agree. The other thing to say is that we've been in discussion with various ethics groups, and describing something as de-identified is an interesting challenge. And there are times when just the fact that vaguely the postcode, the gender, and the diagnosis can enable a clever detective to hone in on a particular group of houses and identify the patient that is being talked about. So I think privacy and security are an ongoing challenge, and I don't think we'll ever get completely secure, but somehow or other we have to work within the environment of acknowledging that little bit of uncertainty and make research work.
Yeah, I suppose there's theoretically, I guess there's possibly re-identifiable, but there's also, is it practical, and can normal people whatever really do? I can't do that, and I work with DNA, but to re-identify somebody. So I think there's there are various levels of risk, as you say, and we have to be as careful as possible. And there's also a little bit of a difference between, I think, maybe germline, what you have in your whole body that could also be in your relatives and so on and so forth, and particular somatic, like the ones that you get from wear and tear, those kind of alterations. So probably less easy to identify somebody from the somatic alterations. And, you know, I think right now a lot of the testing that is being done in the cancer realm currently for clinical practice would be in the realm of panels of specific cancer genes being sequenced and the mutations in those genes, rather than the entire cancer gene, the entire genome of the entire patient, which might be a different level of identification as well. Erin, do you have anything to add to that? It's an issue that lots of people, I mean, you could discuss ethics of genomics
yeah, I would just add that I think we would need an hour and a half to just go through the ethics of data privacy. I would say that if you are going to get this done, it's very much one thing to get it done within the public sector, where they're subject to research ethics boards and that kind of thing. And it's a very different thing to do it with a private company who can resell your data to third parties, perhaps without you knowing it. So that's just something to bear in mind.
Yeah, amongst all the little fine print at the bottom when you sign and submit your sample, is that, yeah, it can be shared. I think the other thing, a lot of the genomic information is complex, and even for people who are in the oncology field, you know, not everybody is really familiar with, you know, what is the meaning of all the different alterations. And sometimes I worry that, you know, information that is sort of commercially available, like that you may not really be able to understand, you know, what is the significance of the findings, and without a genetic counselor there, and, you know, you could sort of go down a big rabbit hole. But, you know, I think those are some of the interesting issues in that private testing domain. I see a few questions that are specific to particular patients or specific diseases and specific clinical scenarios. I'm just going to say, not that it is not important, but I'm not sure that this is the right forum to answer specific patient, you know, dispense specific patient advice. So I think in that, if you have specific questions on your disease or a relative's disease and their treatment, the first place to go is to the oncologist, your oncologist that's taking care of you, or the patient's oncologist. Go to their for that answer and that specific guidance, because it's very hard to dispense medical advice without knowing all the information, and really this is not quite the right place for it. So I'm just going to redirect those questions right now. There's a question to Erin. Erin, is there any research on genome damage from 5G radiation?
Okay, yeah, right. So
I don't even know what that means.
Yeah, so it's what the person there is, I believe, referring to is our cell phone networks, right? So 3G, 4G, 5G. So the short answer is, to this date, with the technology we've had, 3G, 4G, there has yet to be any indication that electromagnetic radiation from our wireless communication platforms causes cancer. And in fact, from the technology up to now that we've had evidence to base research on, 25,000 articles actually says, no, your cell phone and the Wi-Fi networks, at least the doses that we would encounter normally as a civilian, are not going to do anything. 5G is just about to be rolled out. And so I'm not going to say whether it is or isn't going to do anything, because we don't have any evidence upon which to base such a statement. Based upon the initial technological inspections, there's no reason to suspect that it will be any different from what we already have rolled out in terms of overwhelming cancer risk. But like any new thing that's rolled out, you never know until actually you observe what happens in the field.
And I guess one thing that's maybe slightly tangential to that, another thing that has changed recently, okay, the rollout of 5G, that's coming. Something that's happened right now is COVID. So is there anything that has changed in COVID times that might impact your risk of DNA damage or something like that, or risk of getting cancer, or any behavioral changes?
Yeah, absolutely. And so for many of us, not everybody, you know, think about essential workers, but for a large proportion of the global population, particularly in Canada and the developed West, we have shifted from spending a good proportion of our day in our school, our workplace, to being at home. And the way we're behaving within our homes and the amount of time spent within our homes has dramatically increased by as much as a third compared to what it was before. So let's just consider all of the different things in our house that can lead to cancer. I'll just give one example, which I know a lot about because this is close to my heart, is radon gas. And so radon, typically, you won't see it in your workplace because it's subject to occupational health and safety guidelines. Hopefully your employer has taken those seriously and mitigated and tested that building for radon. But you may not have figured out how much radon is in your house. Well, now, for example, in the early part of the pandemic here in the west of Canada, March and April, it was still really cold. You couldn't really go outside. Your heating was on, your windows and doors were closed. Now you're being exposed to 33% more radon if it's in your house than otherwise you might have. So yes, it has changed our exposures in a variety of different ways.
Can I ask you a quick question related to that? Is it possible, we measure radon in the, with the detectors that you place wherever in your house and in there. Can you actually measure radon in the body?
This is a great question, and everybody cross their fingers that I get my grant in a few weeks' time, because this is exactly the type of science that we're doing now at the Charbonneau Cancer Institute. The biomarkers for radon exposure are not known, nor are the gene signatures within cancers caused by radon. But they are out there, and we now have the technology to finally address that. And I'm imagining and hoping that within the next perhaps as soon as 3, but perhaps as long as 5 years, that we will exactly have those answers. These are large projects being conducted by our team at the Charbonneau Cancer Institute in collaboration with Health Canada and Canadian nuclear laboratories.
One for Gwyn. Thank you, Aaron. One for Gwyn. When you say, okay, right now we only have a few percentage of cancers that have these alterations that we can target. What do you see? I mean, kind of crystal ball wise, what do you see for the future? Do you see pecking away at the remaining ones to understand it, or broad types of therapy that would target the rest, or combinations of therapies? Where do you think this precision is going?
That's a fantastic question. So I think there's 2 avenues. I think that little list of potential targets that I outlined at the beginning of the talk is definitely part of the future. One of the things that we need to do is find how many patients with other cancers other than lung have those genetic abnormalities in them, and if they do these new drugs have an effect there? So there's an awful lot of work to be done there. I think the second thing that's emerging is we're getting better at, instead of just looking for 1 or 2 genes in a particular sample, we're doing more and more profiling. So we now have a capability of doing, at a reasonable price, maybe profiling 500 genes in each sample at a time. And what we learn from there is not just about 1 or 2 defects, but we're learning about the mutation burden in general, but also possibly the prevalence of groups of genes involved in specific processes, including, for example, the group of genes related to DNA repair. And I think there's increasing evidence now that there's DNA repair deficiencies that we can see in a variety of cancers, from pancreatic cancer to prostate cancer, as well as breast cancer, where it's been widely recognized, whereby we can do 2 things. We can start using the PARP inhibitors, which are known to be effective in DNA repair deficient tumors, but also possibly put our standard chemotherapies to better use. So I think there's increasing evidence that things like cisplatin that we've been using, not quite willy-nilly, but across a broad spectrum of cancers without really understanding who's going to do very well with that and who's going to suffer terrible side effects. I think this new technology will allow us to be more precise in our use of older drugs. So I think that that will be a dominant theme in the next 3 to 5 years.
Yeah, and I'll just pipe in. I mean, I agree. Like, we can't lose sight of the therapy reduction as a huge benefit either. In the area that I work in, which is pediatric cancer, where you have, let's say, the developing nervous system, and you have lots of side effects, in the CNS. Therapy reduction, if you're not going to benefit, or if you're already going to be a good prognosis cancer, I mean, withholding some therapies is actually very important. What about one thing that we haven't talked about, but we talked about in previous sessions of Late Night Lab, is detection and prevention. Can any of this information, this genetic information, be used to either detect cancer earlier so that we can treat it better or treat it earlier, or I guess be more active in predicting your risk or modifying your behavior? I guess that's to either of you. Can you
I'll speak to that one. A big part of what we're trying to do is not only understand the genetic signature within the tumor that's caused by radon, but who amongst us is the 1 in 30 that's also more at risk. I get a lot of people who test through our national study who come back testing very high for radon exposure. In many cases, that dates back several decades. And they ask me, Dr. Goodarzi, am I going to get cancer? And right now, I have to say I don't know, because we don't fully appreciate who's sensitive. But this is a big thing with exposome data. You need to couple that with genome data, because if once you understand the exposure and the intrinsic risk, you can very effectively direct people to early cancer detection programs. We do have the technology right now to very effectively screen people for lung cancer, hopefully very early stages, but we need to know whether they are at risk. These programs are very expensive to run to our healthcare system, and it's not something we can roll out for everybody in the general public. You need to know not only have you been exposed, but you are also particularly at risk. So yes, we can and we should, but we're not fully there yet for everybody.
And Gwyn, I guess that's risk, and then I guess the other part of the question is detection. So like sort of in the clinic, might you be able to?
Yeah, so early detection is definitely the key to, or a major key to reducing the cancer burden in the population. And probably the flagship programs for that have been cervical screening, which has probably been the most successful screening ever, really. Colonoscopies have been shown to reduce mortality from lung cancer. Mammography is a little bit more controversial, but there seems to be a benefit there in particular age groups. And as Aaron was alluding to, there's now evidence to suggest that low-dose CT scans in certain proportion of patients can be effective in lung cancer. But wouldn't it be good if we could have a blood test that is able to pick them all up at the early stages and tell you exactly where to look to find it? Wouldn't it be great if you could just walk through a scanner like you do at the airport and be told either you're cancer-free or that there is a very, very early cancer in a particular organ? And I think we're still away from that, but there's clearly new technology and new innovation in blood tests in particular that look at particular spectrums of metabolites and even cell-free DNA. This is DNA that's being released in the bloodstream by lots of different cells in the body, including cancer cells. And particularly with these kind of mutations that we've been talking about, it may be possible to detect these earlier in the blood than we can detect cancer on imaging. So yeah, there's a lot of promise and there's a lot of work going on, but there's nothing that we can pinpoint right now that we can tell people, go out and have this test done and you'll know one way or the other tomorrow.
I guess in that sense, I mean, it's very promising. This is why we need research, right? And those questions are out there, and we need matched bloods from people before they have cancer. And there are projects going on right now that are collecting samples, both from patients who don't yet have cancer and might develop cancer in the future, as well as cancer patients, to track that. So, I mean, I think that's a really exciting area. No, we don't have any answers yet, but I think eventually we will. I do see, I'll just comment, I see a few people have raised hands. If you can, instead of raising your hand, type your question in the chat box, that will be helpful. The chat box is should be at the bottom of your screen. It looks like a little word bubble next to the participants. Just type your question there and we'll get to it. Thank you. Let's see. I just want to check with maybe Sandy and Steve if we are until 7:30, or do we have longer? Okay, until I hear that we are ending, I'm going to keep peppering you with questions that we got.
Welcome to keep going, Jen.
Sorry?
You're welcome to keep going.
Okay, thank you. So we talked about privacy, navigating. Yeah, how does our DNA change over time? So why is it, Erin, you showed it this one graph where the percentage was over time, like older and older, there was a bigger percentage of the cancers were in older folks. Is it that our DNA is changing as we age, or what's behind that sort of age rise?
So there's a number of things that happen as we age. One is normal wear and tear on the DNA. Just through breathing oxygen will cause a sort of baseline accumulation of mutations. That's kind of just the cost of doing business as being human beings. Now you can speed that up, or you can somewhat slow that down, again, depending upon your health and your behavior. And so this is one of the reasons why we all may appear to age differently. If you think back to perhaps some of the people you went to high school with, and if you're like me, that was about 20 years ago, more, then it's you see them now, and people look different, and that just speaks to the fundamental differences in the way we age, that is definitely about our DNA. The innate amount of antioxidants that we have in our bodies will suppress some of those mutations. Now, in an aged individual with just normally aged DNA, our capacity to repair new damage that comes in is very different. Typically, a very young cell actually has a little bit greater DNA repair capacity compared to a very old cell. But what's good is that the consequences of new DNA damage actually somewhat goes down as you near the end of your life, where it's actually much greater at the very beginning of your life. And so, yes, age matters. And DNA damage is a very important factor when you consider how you individually are going to age, but also what your risks of new exposures might be for your kids and grandkids compared to your grandparents.
Thank you, Erin. Actually, I did not know that young cells can repair damage better than old cells.
It depends on the type of damage, but for example, the break in the DNA double helix, a full break, yeah, we have evidence, and it's been published, that if you are an aged cell, they have less capacity to cope with that versus a young cell.
Okay. Well, I think we should probably wrap it up soon, or actually maybe now. There's been lots of great questions, and I appreciate it that people had asked for topics in the future. So just to reiterate how these sessions go, we try to pick a couple of theme questions to go around. We can't cover all of cancer in an hour or less. But we try to pick focus areas that people are interested in. So thank you for suggesting some future topics for this late night lab. And thank you, Gwyn Bebb, and thank you, Erin Godarzi, for being our guests for the evening and our experts. I learned lots of stuff too. And then finally, I will make a plug for research in general. Of course, none of this progress that we have made, at all, is without research, right? Even the original chemotherapy, that was not without research. But this really exciting era of precision therapy, molecularly targeted and molecularly informed therapy is really not possible without research. And if you're in a position to contribute to research, even as a participant in a clinical trial, so if you're in that category, please do. I mean, this is how we make progress. And finally, the two folks that we've had on today are part of the Charbonneau Cancer Institute, the University of Calgary, and the health system here in Calgary. So these are your local heroes. So thank you so much, and thanks for joining, guys. We'll see you next time. You'll get an email.
Fantastic hosting.
Thank you. And thank you, CPL.
Thank you, Dr. Chan. Thank you so much, Dr. Godarzi, Dr. Bebb, and Dr. Chan for this informative and engaging evening. Thank you to all of our presentation partners, the University of Calgary, Alberta Health Services, the Tom Baker Cancer Centre, and the Arnie Charbonneau Cancer Institute. Thank you to our incredible staff and to each of you for attending this online discussion. You will be asked to complete a short survey by email, and I invite you to kindly share your thoughts to help us improve and plan our future programming. We look forward to seeing you again soon at another presentation. Thank you.